No, I have to. I have to stand the whole time. So since we had limited questions before, I would just say, are there any, deeper questions that people want to dive into? We had limited questions just for clarification, but does anyone have any specific questions for any of the presenters? Then I guess for each of the individual presenters, just if you could talk a little bit about the qualification process itself, except perhaps for Ann, because you're not putting anything in, but where you see the concept that you presented, where you are in the step going forward with qualification, and how you think it would be of value. Yeah, Eric, maybe if you.
Well, we're early in the process with the mouse. I think the value for any of these on the most basic level is an opportunity to gain greater insight into how the model may be used, and I think it's certainly the case for many of these animal models that there's a fair amount of uncertainty, let's say, that's propagated by various proponents of various models, and it may seem to people interested in evaluating new drugs or new regimens, but potentially new sponsors to the area, that perhaps there's not much consensus on whether the mouse is useful at all, whether you have to go to a larger animal, what can be learned from these various species. And so I think at its most basic level, this is an initial step to try and understand what each model can contribute. And I think a real solid opportunity to ferret out the positives and the negatives. I think we'll find many cases where in the case of the mouse, that it has performed quite well, but it has obvious potential limitations that have to be examined.
Bob?
Well, maybe I should just take a minute, and after having said how wonderful 2-month culture is, I should talk about its limitations. I mean, it really, the confidence intervals are wide. In order to get to a 4-month regimen, we think pretty much you have to get down to 1% positive at month 2. I think we probably will close in on that target relatively soon, at least for fully drug-susceptible TB. And that's about the limit of information that you're going to get from a phase 2 study. You really can't discern rates lower than 1% without doing what the equivalent would be of a phase 3, a very large phase 2 study. So it becomes rather meaningless. And that's why these other culture-related endpoints can become so important. So a time to events or LAM or other types of markers. But given this opportunity, given what we know about month-through culture, and given the opportunity and the need at the moment for the pan-TB regimen, I think there is a real opportunity to use it while we can.
So with the PBPK models and the qualification, I guess, as I explained, we're still fairly early. We've had an initial telecon and an initial face-to-face meeting. And I think it's interesting as well, it's in the steps we're taking to qualify the PBPK model around TB are in a wider context where there's a lot of regulatory interest in a minute about the qualification of PBPK models in general. So as I was explaining with the EMEA bringing out draft guidance, and then the feedback that's going on with that guidance that will be formalized, there will be some probably a need, particularly for platform models, to be undergo some qualification. So I think what we're doing in the context of the TB model is, you know, fairly early. I think we're one of the probably the first case that we've had an actual face-to-face discussion with the agency about qualification of PBPK model. But I think it's going to become kind of standard practice anyway as we go forward.
And so, and I don't know if any of you could comment on it, but just how you see the work that you're doing and how that would interact with, or how it might build on, you know, say with the mice and the PBPK or the hollow fiber system models, and how that all might, you know, relate as a broader context for either advancing drugs into clinical development or not advancing drugs into clinical development.
See, as I'm holding the microphone, I guess I can have a go first. I think the way I see it with the, you know, there's a lot of different types of modeling initiatives that are going on, and really they're a way of integrating information so that you can try and make the best decisions. So you've got information around, from hollow fiber, around how effective certain concentrations are at killing bacteria in certain different scenarios. The PBPK models are helping you to try and understand whether you actually ever see that concentration at the site of action you care about in the body. So it kind of integrates all of the information you have about the drug fairly early on to try and help you make better decisions about whether you want to carry on with that drug or switch to another one, what kind of study you want to do, what kind of dose level you want to use. And particularly drug interactions, which are always going to be a problem with combinations. There's really a lot of information that you can gain from modeling to understand whether you're going to have a potential risk or not. That you still might ultimately confirm in the study, but it helps you to make those decisions early.
Yeah, I mean, I think with respect to the animal models and the hollow fiber model, as Ian mentioned, I think the you know, the issue is the translatability. I mean, what we all want is to be able to evaluate new drug regimens in a series of models that you know, really hopefully beginning, for example, in a hollow fiber system, one can define PK/PD relationships that one can then confirm in an in vivo model. So there you've perhaps reduced the amount of animal experimentation that you would have to do, and then hopefully by doing more animal experimentation, you have enabled translational models like we heard of from Radha and her group this morning to begin to integrate the PBPK, the different subpopulations, whether these are empirically defined or more rigorously defined by the culture conditions in the hollow fiber system, for example, and you know, begin to get greater insight into how dose optimization and specific drugs might be selected as combinations in an effort to you know, limit the number of trials that have to be done. So you know, I think that all the, you know, the extent to which we can relate concentration at site of infection to effect and show the portability or the translatability from model to model, that's you know, that's part of the process of gaining confidence and that you've got the right dose and right combination.
And I'll just reiterate what I mentioned from a regulatory perspective. I think what the qualification process does across all of these programs that are in different stages is to really give you a framework of developing data that are closer to being regulatory ready for whatever use you want to use them for. And I think that is a very valuable aspect of qualification, the qualification process, regardless of where you end up.
Andy, you have a question or comment?
Two questions. One is a quick one for Eric. You mentioned the review that's underway by the working group on starting with the BALB/c mice. What's the timeline for that? Did you mention that, or I'm not sure I heard that?
No, I didn't. We're in a process, I guess, of, we're early enough in the process that we have, we developed an initial template for the data transfer to be able to take the data from the various research groups that have, compiled data over the years to put it into a format that can then go into the CPATH platform so that it can be ultimately be compared to clinical data. So, I think we're getting to a point where we can define the right structure of that template, hopefully within the next few months. We've already started to input some of the data, specifically from the Remox regimens, but it's in and of itself an iterative process. So, I think we could probably, I don't think it would be stretching it to say that we could probably complete that for some of the basic, the Remox regimens, for example, and incorporation of rifampin and pyrazinamide into first-line regimens, which are our priorities, and have an initial statistical analysis within the next year.
Okay.
That's
you got real quiet there at the end, but I heard it. And then I had a question for Bob, and that was, we have from the experience now with MOXIE, at least the knowledge that we can see what appears to be, at least in some settings, a promising EBA that doesn't correlate with a promising sterilizing ability. Do you know of any reason we should be concerned about the reverse? That we could see what looks like a good EBA and actually it doesn't sterilize?
Yeah, I mean, I think isoniazid is the typical example. Obviously, under some circumstances, it does kill quiescent organisms. You can prevent TB with it. But you could mistake a poor drug for an excellent drug and the other way around.
So INH would really have failed us right now if we got it tomorrow again.
In a number of ways.
Yeah, so there is an important lesson, I think, that for all of these biomarkers is that it's so important to have as robust data connecting to a clinical endpoint as is possible, because there are very powerful forces, whether this is an academic consortium or a pharmaceutical company, that either will want to move the drug ahead or not move it ahead at all, that have nothing to do with the likelihood of success. And we need to be able to speak to that with hard data. And this is exactly the sort of setting that can generate that data.
Were you trying to make a resounding statement of support for the long-standing criteria FDA has used?
I'm not...
By your emphasis on the importance of the clinical endpoint.
Yes, yes. It's very important. And I didn't mean to, in my presentation, I didn't mean to negate that. But the question is, how do you get there?
Klaus?
Yeah, thanks. I was just going to ask Anne if you could elaborate a little bit more on this other pathway that FDA has dedicated specifically to quantitative drug development platforms, like the PBPK model and the liquid culture model, which is this fit-for-purpose initiative, which is reserved specifically to these kinds of quantitative models. It's not called qualification, it's called fit-for-purpose, but it's very much equivalent in its steps to the typical qualification effort for biomarkers and outcome measures. So I wonder if you could elaborate a little bit more on that one.
I could, but you can stand there because you can do it better, because Klaus has been doing this. But as he said, it became pulled apart at you actually do know the history better than I do, Klaus. But there were some work that we were doing that CPATH was doing that wasn't considered qualified, but it was fitting a purpose, and they were afraid to qualify it because they felt like it was going to be changing a lot, and so but they still saw the value in it, and so that they very recently, I would say, in the past year, is when they've really laid out the fit for purpose steps for that. But it is what can be used for these types of modeling and simulation that you know there's going to be a lot of changes. But again, similar steps, as Klaus said. And really, Klaus, if there's more details you want to fill into that, I know it's something you've been working with even closer than I have.
The process is very much similar in the sense that you start with a context of use and then you follow a series of steps and submissions and face-to-face meetings. But the important thing to keep in mind is that just because you kill something one name doesn't necessarily mean that that's the only way to get regulatory endorsement. I think what we are really after through these kinds of collaborative efforts is really regulatory endorsement for the kinds of solutions for the drug development process that we're thinking about. And qualification is certainly a very valuable one, but it's not necessarily the only one. Just to put another example on the table, the way that FDA was open to including the whole fiber system submission as part of the docket for the drug development guidance is another form of very significant endorsement. So just so that we know that what we really want is to get that level of endorsement so that the tools can be used in confidence by the drug developers, Larry and Bob and the TB Alliance, et cetera.
Right, and again, that's why I think this process is so useful, because no matter what your endpoint is, that is what we want. Even if it ends up that one individual company wants to take this and go through their own drug development pathway with it, having these data to draw upon, whether it comes from a guidance, whether it comes from qualification, whether it comes from a presentation that you do at a workshop, I think that's what we're aiming for. Yep.
Thank you. Any other questions from the panel or comments to the panel? Going once, going twice? Then we're adjourned, and we have the reception at 7? At 7 in the little ballroom upstairs. Thank you very much.